quick video over mitotic, kind of the evolution of it, but more important, the regulation of it. A couple things is that obviously prokaryotes were first, and that we believe that the evolution of mitosis would have came from our ancestors that are bacteria. They do a form of binary fission, which is simply a splitting and dividing. Primitive organisms like these dinophalanges, which are our protist, have a slightly more complex form, but still pretty simple. You can see their microtubules attached to the cell membrane. And then we start getting a little bit more complicated as we go down to our current version of the process. And it really, again, points to the evolution, considering that these two middle protists, an intermediate between us and bacteria, do kind of a quasi-form of binary fission. It's a regulated, it's a system, and so what we see here is that there's certain cells like neurons and muscle cells will not divide after functioning maturity, so they'll stay in that G sub 0 stage, while stem cells, they'll actually divide pretty quickly, specifically in the bone marrow, which will make blood cells, which die at a really, relatively speaking, quick pace. Red blood cells will only live about 120 days, but if you think about their actual maturity, they don't last very long once they don't have the nucleus, and so therefore reproduce rather quickly. There's some evidence that the cytoplasm is what's responsible for telling the cell to divide, and I'm not going to get too much into this, but if you look at this diagram here, it's easy to explain. So you can see the S is the S stage of this process, and so what's happening here is when we look at these different cells that are going on here in the divisionary process, if you look at the two different ones, so the cell cycle control system, we're directed by, it's like a clock. I use the example in class of a washing machine knob, and there are those checks and checkpoints in there that we need to hit and go through, and there's certain things that must be achieved. So we're going to talk about those. Most of this already covered in class, but I want to kind of hammer home a couple points. So there you can kind of see the three big checkpoints there are in red. One happens late G sub 1 prior to S stage. Then we have the one at the end of G sub 2 and one right there in the middle of mitotic division. The G sub 1 is the most important. We talked about why that is. It's a checkpoint that allows us to kind of send it to G sub 0. And then at that point, if we go through that checkpoint to the S stage, we're pretty much guaranteed the cell has to divide. If it doesn't, then it will go to that G sub 0 where it gets fixed or have some corrections made for the cell. Again, this kind of just shows the checkpoint being up. In this case, goes to G sub 0, which is a state of non-dividing cell just doing its function, just a stage of suspended animation. That just kind of shows that the variation of the G sub 0 will depend upon the cell itself and what the cell's doing. Some cells, as I mentioned earlier, like nerve cells and muscle cells, will stay in that stage for the remainder of their time. There are two things that will be regulating, and this is going to be important: cyclins and then the CDKs, the cyclin-dependent kinesis. Those are both proteins that are heavily involved in this process. And the reason we can kind of see or tell this is that these two proteins, cyclin and CDKs, will actually fluctuate during different times of the cell process. So when we're looking at cells in different stages of mitotic or cell division, we can account for different amounts of these two chemicals, these two proteins. So the diagram here kind of shows during mitosis, you can see how both the MPF is increasing. We'll talk about what that is in the cyclin concentration. It's growing starting in S up through G sub 2, and then it has its peak right in the middle of mitosis, and then it steeply drops down until S stage again, where it starts to increase, and it will continue in that process. The MPF, on the other hand, which we'll talk about, will increase specifically during the mitotic cell division time period. So what is MPF? Well, the MPF has to do with the fact that when the CDKs and the cyclins, those two proteins, come together, when they come together, they form this MPF molecule. And what ends up happening is it's during its highest amount, like on the previous slide, right there during the mitotic division. The cyclin itself will break down, disintegrate, therefore leaving the CDK. It'll start to increase again. The cyclin will start to increase again, starting in S, increase stimulating the cell process. So basically what this tells us is the amount of cyclin and the CDKs will kind of be responsible for determining the rate of these reactions. What could cause a signal at a checkpoint to be stopped? The kinetochores are the motor proteins that are going to attach from the microtubules onto the centromere of the chromosome. And basically what happens is if that does not occur, and the microtubules do not, they'll send a molecular signal. That's molecular signal during the delay anaphase, so it causes a delay between metaphase and anaphase. And that's what really the cell's waiting for during that time, to make sure they're all attached. There can also be external signals that can cause delays in this process. So proteins released by other cells can cause cells to divide. In other words, a nearby cell, maybe as it's dying, will release some of these proteins to stimulate nearby cells to divide to fill in the gap for the cells near it. One of these particular protein factors is called PDGF, platelet-derived growth factor, and that's responsible for increasing the number of cells in a particular culture. And so as this chemical is released, it causes the cells to divide. We can just simply add that to a culture of cells, increasing the likelihood that cell division occurs. You can kind of see the example here is we have a bunch of cells in a petri dish in a container. We add the PDGF to it, okay, and we start getting this growth that'll occur, and we get this rapid amount of cells that divides and fills up the container. Okay, so we put it on there, all of a sudden we see this increase in growth filling in the gap. So it's an external chemical that releases, that increases the likelihood. Density-dependent inhibitors are basically the idea that cells will stop growing when they're next to and fill into another one. So if you can think of like a dish, if one cell forms, another one next to it, it won't need to divide. So it basically the density of the cells around it prevent it from growing. You can kind of see this here when you have a full slate of cells. If you take a couple out, they will be filled in by nearby cells to meet that density dependent factor, but then they stop growing. The issue is when they don't determine those factors, and then they'll keep growing anyways. There's nowhere to grow this way or that way, so it grows up, forming what we call a tumor, and that's where cancer can come into play. Those cancer cells keep dividing rapidly, and they aren't functioning properly, and they'll basically choke out the remainder of the cells nearby. So again, cancer cells will not exhibit either density dependent or the anchoring dependence, and therefore those cells will not, as you can kind of see in that bottom picture there, will not, they'll continue to grow, they'll grow vertically, they'll create a tumor, and then they're not anchored, so that doesn't stop them. They can leave the area that they're initially found and can spread as well. Here's a short video that kind of goes into that. So you just kind of see, again, those cells that are experiencing cancer, they don't have that anchoring. The reason cancer cells develop is they do not have the normal checks and balances. They blow right through the checkpoints. They form tumors, and then A couple key terms you might want to know is malignant tumor will spread and can metastasize. Metastasize is when it breaks apart and goes to other parts of the body. So that's kind of when it gets really bad. If a cancer cell, let's say, in your skin metastasizes and spreads to your lungs, you can have some major, obviously, issues in the body. So those are just some key terms that you'll need to know over cancer cells for the test, what they mean and what they're responsible for. So make sure you write those down or go over those. I'm going to kind of move on to the next slide. So if you need to pause the video, this would be a good time to do that. And you can kind of just see here the different stages of that. So that's all I got. That's kind of a little bit additional into what you read into 10.3. So make sure if you have any questions, bring them to class. Thanks.